Champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days because we have a very structured plan and I'm not kidding. So today we are going to deal with innate and acquired immunity. This will be video number 14 and going ahead, why we chose this chapter, because in the previous years, the number of questions have been increasing. So we can expect the same for this year as well. So it has been 1, then it became 2, 3, 4. This year probably 5, or it could be back to 3, we never know, but it's our duty to study it, so we have to do it anyways. Yeah. And what did we cover in the previous session, children? We spoke about helminthic disease, that is elephantiasis and all, and ascariasis, and then we spoke about fungal disease, that is ringworm, and also I spoke a bit about bacterial, viral and protozoan diseases, which we covered obviously before doing this particular topic as well. So if you want to see those videos, you are most welcome. The videos are there free for you. You can watch it anytime. Now if you want the PDF for the same, that is all my notes, you can join the WhatsApp channel as well. I don't want to waste your time, so let's go ahead. So over here, were you able to solve this question? I was really happy. Inchara solved this in the comment section itself. I was very happy, and then I saw someone else also. I remember Krishna is the name. I'm not able to bring back the memory, but I was really happy seeing you people commenting down the answers, because that means you are participating in my class just like how I am participating to give my best for you guys as well. Yeah. So common cold is not cured by antibiotics, because why is common cold not cured by antibiotics? Why does not a doctor give me an antibiotic when I go to him or her if saying that I have a common cold? Because antibiotics are for treating bacterial diseases, children. The diseases that are caused by bacteria, that is cured by antibiotics. Well, you are talking about common cold. Common cold is caused by a virus. What virus? If you remember, a group of viruses which are known as what? Rhinoviruses. And why is no one able to solve this trouble of having common cold? Why is it not treatable? Because these group of viruses, that is rhinoviruses, they change their genetic makeup. For example, I bring out a cure for it. The second moment they will change their genetic makeup and be something new, so that this particular treatment that I found for them won't be useful for it. You get it? So this is how fluctuating their genetic makeup is, which is why we are not able to find anything against them. So that is all about common cold, and this came in the year 2011. Yeah. And now let's start today's topic, that is immunity. We'll be talking about innate and acquired. What do you mean by immunity? That is the first question. So when I say that I am immune to diseases, that means my body is able to fight against all the diseases that comes through my way. Right? So in short, the statement is the overall ability of the host means me, where the pathogen is going to live, to fight the disease causing organisms is known as immunity. Now immunity again can be divided into two types. One is innate and the other one is acquired. So as you understand from the term, at least if you do not understand anything from innate, it's fine. Acquired means something you are acquiring. Okay, you're acquiring in your lifetime. This is not something that is inborn in you. You are acquiring through your lifetime. This type of immunity comes throughout your lifetime, and you will get to know about that really soon. As of now, we have to talk about innate immunity. So innate immunity is inborn. It is within us, and if you see the examples, you will get to know. So skin is a type of innate immunity, or basically the secretions we have in our body is a type of innate immunity. It is there with us throughout birth. Okay, from the time we are born till now, it's there with us. Now this type of immunity is non-specific, as in tomorrow if a virus comes or a bacteria comes, it won't do partiality and say that I would act on you but not on you. It's not like that. It's not specific. It can act on anybody. Okay, it is so impartial, you can remember like that. Now it is present at the time of birth as well. So you have to remember these two very important things that I have written over here. Innate is obviously always non-specific. It is not specific. It can act on anything. Now you'll get to know. So over here, the first example of innate immunity is physical barrier. Okay, what is it? Physical barrier. So what is acting as a physical barrier in our body? Obviously the first physical barrier is the skin. So skin, have you seen that skin says that, hey, bacteria, you can get inside, virus, please stop. It's not like that. It acts on both. It says, please, bacteria and virus, do not come inside. This is not your place to stay. So basically skin is one example. It is acting as a strong physical barrier. Now skin have hair as well, which is again acting as a barrier because it traps all the organisms. Okay. Now Now. Mucus, you know, inside our body there is a layer which is the innermost layer, children. Okay, so just imagine that this is the inner side of our body, probably our neck region or whatever. Okay, alimentary canal, let's take alimentary canal. Okay, so now the innermost region is known as mucosa layer. It is known as mucosa layer, which is actively having cells that releases what? Mucus. Now you know how slimy mucus is, and what does this mucus do, children? Mucus will obviously trap what? Antigens or pathogens. Now it is common for all, it can trap anything that gets inside our body. So both of them, they are acting as a physical barrier. They're not letting to cross this barrier and get inside our body and cause an attack. So that is why they were categorized under physical barrier. Now do not put skin and mucus under physiological barrier, that will be something else. Okay, so they are acting as a strong physical barrier, which is why they are put in this particular category. Going on, second, we have physiological barrier. Okay, something that our body does and which is common for all, it is it's destruction for all. What is that? First thing, secretions. Our body give out various secretions. For example, if you know our stomach itself, if you see our stomach is responsible for secreting what? A very strong chemical, which is HCl, hydrochloric acid. Now what does hydrochloric acid do, children? Hydrochloric acid goes and works on the pathogens and kills them. Obviously, can any pathogen live inside an acidic medium? No. Most of them, they do not. Some of them, they could and they survive as well. Okay, but most of them, they cannot survive in an acidic medium. Now, who is responsible for producing HCl? It is the stomach. What is there in the stomach that produces HCl? If you know that, I'll tell you. If you do not know that, I can tell you. In the stomach, we have a cell which is known as oxyntic cell. Okay, which is responsible for releasing HCl. Now, oxyntic cell is also known as parietal cell. So, you can remember ox is very dangerous. So, similarly, HCl is also very dangerous. Okay, so this particular portion comes in when you are studying in 11th for digestion and everything. There, this particular portion comes in. As of now, you don't have to just remember this particular thing that I have shown over here. Okay, just remember this thing, that stomach has a secretion which is known as HCl, and HCl is something that provides an acidic medium for the germs to get killed. Okay, now apart from our stomach, you know, when we cry, we produce tears, right? Apart from tears, in our body, we have salivation, where we produce saliva, right? So what is common between tears and saliva? Tears and saliva, now children, they are having an enzyme which is known as lysozyme. What is it, children? Lysozyme. And this lysozyme goes and acts on the cell wall of the bacteria. It goes and acts on the cell wall of the bacteria and kills the whole bacteria. Okay, now depending on whatever pathogen it is acting, but it has different ways. But usually if it is a bacteria, it is going to act on the cell wall. Okay, what is the enzyme? Lysozyme. Where is it found? In the tears as well as in the saliva. Okay, so all these things that you see over here, that is HCl, tears, saliva, all of them, they come under this category, that is secretion, and it is a physiological factor because it is being produced in our body. Okay, now the next one is fever and pH. So in our body, we know when something comes in, we have very high temperature. Now why do you think there is very high temperature? In this high temperature, do you think any organism can survive peacefully? No. For example, if wherever you are sitting as of now, you switch off the fan, okay, or you put on heater, okay, either way you won't be able to properly sit there. Why? Because it is not an optimum temperature for you. Similarly, in our body, when fever comes, there is high temperature, which is why lots and lots of pathogens die. Okay, now apart from that, pH is also one more thing. If a pH is disturbed, okay, if a pH is disturbed, obviously not every pathogen can survive in all the pH. They also have a proper pH to survive. So now again, what is important here? Please remember secretions here. Secretions are very much important. Please remember lysozyme here. Okay, so physical and physiological barrier is done. Coming ahead, talking about cellular barrier, that means now cell is going to do something. Certain types of cells, they are going to act as a barrier. They are going to protect our body from something. Let's see who are those cells. So we have phagocytosis cells. What do you mean by phagocytosis, children? Phagocytosis, you might have heard about that, right? When something is taken inside the body and being broken down, this is known as phagocytosis. Okay. Now penocytosis is when something liquid is being taken in. So as of now, something solid is being taken in and broken inside the body. You might have seen in amoeba and all. Amoeba taking their food, goes into the food vacuole, and the food vacuole, the food is phagocytized. It is getting broken down, right? Similarly, over here inside our body, there are certain cells, which, for example, just imagine there is a cell over here, okay, and there is a pathogen over here. So this particular cell will slowly engulf this pathogen inside it and then later kill it. So these type of cells, they are known as phagocytic cells. Okay, it is known as phagocytic cells. Now what are the examples? In our body we have neutrophils, very much important, please remember. We have neutrophils, I'll tell you about neutrophils, and also we have one more thing, that is what? Monocytes. So neutrophils and monocytes both are phagocytic in nature. Okay, so you might be thinking, why are both required? If one cell is there, that's the only thing, right? Now I'll tell you. Neutrophils are comparatively smaller. Okay, neutrophils that you see over here, they're comparatively smaller. But monocytes, no, monocytes are there, and what happens to monocyte is that it is present in the blood, but now slowly when it gets into the tissues, it gets in between cells, it transforms into something that is known as macrophage. What is it? Macrophage. And now this macrophage, they are comparatively what? Bigger children. So neutrophils smaller, macrophage bigger. Now neutrophils have a different name as well. They have one more very important name, that is polymorphonuclear leukocytes. What is it? Polymorphonuclear leukocyte. Why? Children, when you talk about a cell obviously has nucleus. Now in their nucleus, the morphology is different, the appearance is different. Basically, they might have multiple lobed nucleus, which is why they are known as poly as in different, multiple, morpho as in morphology, nucleus as in nucleo as in nucleus, and leukocyte because they are all white blood cells. So over here, monocytes, neutrophils, all of them, they are coming under this category, that is white blood cells. You know, in our body there are three types of cells: red blood cells, okay, that carries hemoglobin for oxygen transport; white blood cells which fights against diseases. There are different number of white blood cells. Two of them are here itself, okay. Apart from that, there are lymphocytes and all of them. I'll talk about that. And then there are thrombocytes. Thrombocytes are what? Platelets, which helps in clotting of the blood. These are the three different categories. Now we are talking about the second category, leukocyte. Inside leukocyte comes these both, okay. So neutrophils and macrophages, they are both phagocytic. Okay, what is the difference? Here the nucleus is kind of different, which is why it is known as polymorphonuclear leukocytes, and macrophages are bigger in nature. Now neutrophils are the one, for example, one pathogen comes, the first person to go there would be a neutrophil. If you compare between them, the first person to go there will be a neutrophil. So I'll give first category, then neutrophil calls like, hey macrophages, come, you also come. Second come macrophages, and they now destruct everything which is causing a trouble for our body as well. Okay, will you be able to remember this? It is easy. Okay, it is easy. Now talking about non-phagocytic cells. Non-phagocytic cells as in they are still a cell, they are acting as a barrier, but they are not doing phagocytosis, they are not taking something inside. So example of this is natural killer cells. Okay, natural killer cells, as in they are naturally going to kill cells. How? So basically, natural killer cells are responsible for a very important secretion which is known as perforin. Perforin. Now with the help of this name, you might understand it is going to cause perforation of something. Okay, perforation as in making holes in something. For example, there is a cell over here. Now this perforin and this particular cell is basically a pathogenic cell. Or not a cell, basically an organism. Just imagine that this is an organism. Okay. Now this organism, when perforin is being released by natural killer cells, it goes and perforates the wall of this particular organism such that whatever water, whatever things are there, it could get inside the cell, it could get inside this organism, and basically kill this organism. It can be anything that gets inside. Okay, basically the water can get inside and make the cell swollen, make the organism swollen and let it die like that. But the major thing is that it releases perforin, and this perforin will make pores, and later after that after making pores, obviously there are lots of ways to get killed. Okay. Now that is about natural killer cells as well. Now this is also a leukocyte, this is also a WBC. Now before going ahead, I want to talk a bit about WBC because I know you might be having a little amount of confusion in here. So this particular slide I have you have to take a screenshot because this won't be there in the PDF. This has something that I have added. Okay, so when you talk about WBC, no children, they are also known as leukocytes. Okay, now there are two. One is agranulocytes and the other one is granulocytes. Okay, agranulocytes, as in it was said that they do not have granules in which enzymes and everything is there, but in their structure they have granules which has all the enzymes. But actually it is not true. Initially we did not have good microscopes to look around it. They also have granules. Okay, so basically in their cytoplasm they have granules. Anyways, under agranulocytes come two people. One is lymphocytes. Under lymphocyte comes two, B and T. Okay, B and T lymphocytes. Now going ahead, apart from lymphocytes, we have monocytes. These monocytes, when they get into tissues, they become macrophage. Okay, now talking about granulocytes, we have eosinophil. This cause, okay, what is cause I'll tell you, or should I tell it now also? Okay, they are the ones that is responsible for allergic reactions. Okay, macrophages, as I said, they are responsible for phagocytosis. Okay, B lymphocytes are responsible for making antibodies. Okay, T lymphocytes are responsible for killing cells or helping the other cells. Okay, now apart from eosinophil, we have basophil. Which is majorly responsible for inflammation. Why do you need inflammation, children? When we say inflammation has happened, basically you are dilating the blood vessels so that other cells can easily reach at this particular point. Now you only think there is a small route and there is a big bus. Can it travel through the small route? No. At this point of time, if the route was bigger, then probably the buses could easily travel. Similarly, basophil is responsible for inflammation whenever needed. Okay. Now going ahead, we have one more thing. Neutrophils. Okay. Neutrophils are again responsible for phagocytosis. Now, in between them, one more cell comes in which is known as natural killer cells, about which we spoke now. Okay, so when we were talking about cellular barrier and when we were talking about phagocytic cellular barrier, we spoke about monocytes and neutrophils because both of them, they were responsible for phagocytosis, but who usually comes first? It is neutrophils. Neutrophils are also known as polymorphonuclear leukocytes because they have, I will write it for you, polymorphonuclear leukocytes. Okay, because their nucleus has multiple lobes. Fine. Now, what? Yeah, rest all things are behind. Yeah. So these are phagocytic cellular barriers and this is non-phagocytic cellular barrier. Rest all of them you will be getting to know about them when we talk about acquired immunity. Okay, as of now they are not involved. All of them are involved in innate immunity as well. 1, 2, 3. Are involved in innate immunity as well. That means they might be involved in acquired, but definitely in innate. You can take a screenshot of this. I will move aside. Okay. Now, so over here I spoke about that. Okay, that is a cellular barrier. Now cytokine barrier. What do you mean by cytokine? That you have to understand first. Cytokine means when a cell is going to release something, they are going to, and these release substances are going to cause trouble somewhere else. That is what you mean by cytokine. Okay, when your cell itself is going to release something, and that something could probably help you another way by destroying the other cells. So basically interferons are one such cytokines, and interferons are definitely being released by a virus infected cell. You have to remember it. So virus infected cells, no, they release something that is known as interferons. And why are these interferons released? For making other cells aware that, okay, this cell is virus infected, probably you should be releasing something antiviral so that you can protect yourself. Okay, that is, this particular cell is a good friend that everyone has, right? And it says to other cells that please release what this interference goes and acts on other cells and says release some antiviral things so that you can be protected. Okay, that is the use of interferons. So this interferon is acting like a cytokine. It is being released by some cell. What cell? Virus infected cell. You have to remember this. The questions have usually been framed. So if we talk about innate immunity, that's all. So innate immunity, children, it is non-specific. Okay, anything can be stopped with the help of innate immunity, and it comes from by birth. Okay, it comes with us. It comes with us by birth itself. So it's inborn. Yeah. Now there are 4 barriers: physical, physiological, cellular, and cytokine barrier. Physical, skin and mucus. Okay, physiological secretions, we have HCl secreted by our stomach, and then we have tears and saliva, in which we have lysozyme enzyme, and this lysozyme enzyme goes and cleaves or attacks all the bacteria, whatever is there, and kills them. Yeah. And now when we talk about cellular barrier, we have phagocytic and non-phagocytic. In phagocytic, neutrophils, monocytes, PMNL, name is very much important. Natural killer cells are non-phagocytic and they release perforins. This perforin goes and cause perforation in the cellular nature, in the cell, okay, or in the organism's body. Okay. Now obviously this organism will also be obviously made up of a cell or something. It might also have a barrier that is going to make a pore in that barrier. Okay. Now talking about cytokine barriers, basically the viral infected cells release some chemicals which are known as interferons, and these interferons basically goes and act on other cells, by which we are making other cell aware that something has happened, and you have to be remaining cautious about it. Okay. That's all. Now let's read it once very fast. Okay. Innate immunity is non-specific type of defense, again, okay, that is present at the time of birth. Okay, this is accomplished by providing different types of barrier to the entry of foreign agents into our body. Innate immunity consists of 4 types. Let's see. Physical barrier. Skin on our body is the main barrier which prevents entry of microorganisms. Agree. Mucus coating of the epithelium lining, the respiratory, gastrointestinal and urogenital tracts also help in trapping microbes against entering the body. So what do you mean by respiratory tract? Obviously the lungs and everything. It also has a layer which is known as mucosa. Okay, going ahead, we have gastrointestinal, that means your stomach and everything again has a layer. Innermost layer will be again mucosa. Okay, talking about urogenital, that means urine and genital tract. Okay, for example, vagina in females, in the case of males it is urethra. All of them will be having something so that they could trap the organisms easily. Okay, so respiratory, gastrointestinal and urogenital tract. In there, what do we have? We have mucus coating of the epithelial lining. Epithelial lining is in the innermost lining that is over there. Now going ahead, yeah, physiological barriers. Acid in the stomach, saliva in the mouth, tears from the eyes all prevent microbial growth. How, children? By the release of something that is known as lysozyme. Going ahead, cellular barriers. Certain types of leukocyte, that is WBC of our body, like polymorphonucleoleucocyte, PMNL, neutrophils. Why they are known as neutrophils? Because they can be visible with the help of neutral dyes. Okay. Now, and monocytes and natural killer cells in the blood as well as macrophages in the tissue can phagocytize and destroy the microbes. So remember that monocytes are present in the blood. When these monocytes move into tissue, they transform into something that is known as macrophages, and they are responsible for phagocytosis and destruction. Yeah. Cytokine barriers. Next one. Virus infected cells secrete proteins that is known as interferons, which protect non-infected cells from further viral infection. Yeah. And this is the question that we have got. Tell me the answer for this question really fast. Interferons are synthesized in response to. This came in 2001. It's a very simple question if now you know the concept. Tell me the answer, children, in the comment section or in the chat box, anywhere, please. So it will be what? Viruses. We just studied it. We don't even have to look for the other options as well. Yeah. Okay, going ahead, let's talk about acquired immunity. Now, acquired immunity is like personal tuitions, where, for example, if I know this person, that is X or Y person, I might have a different strategy for this X person, and I might have a different strategy for the Y person as well. So my strategy will be specific to that person. Over here, acquired immunity is pathogen specific. For example, if some pathogen comes, I will be releasing something else. Or if some other pathogen comes, my body will be releasing something else. This is what you mean by pathogen specific immunity. And also one more thing, it develops memory. For example, I'll remember, okay, I'll again remember that, okay, this is something that I have to tackle with. Okay, just forget everything. I'll tell once more. Listen to me very carefully. Once a disease comes in my body, okay, once a pathogen comes in my body and it's like, I'll be causing a ruckus inside my body. At this point of time, the second time when this particular same antigen tries to come inside my body will say, hey, I remember you. This is how I tackled you previously. I could not be very much efficient previous time, but this time I'm very sure what to do about it. So this is what memory is. Our body will remember what kind of antigen has come before. And what do you mean by antigen? Antigen is that something which is going to excite which is going to initiate an immune response. Antigen is the pathogen. Antigen is basically the foreign agent. You can remember like that. Antigen, when I say antigen, it's a foreign agent that comes inside our body and is going to initiate an immune response. Okay, it is going to initiate, children, an immune response. So as of now, when I am saying it is pathogen specific immunity, that means it is our body will be knowing about each of the pathogens or the antigens, and they will be showing specific immunity to that. Okay. Next thing, it develops memory. So next time an antigen or pathogen comes inside our body will be aware that this has already come. Okay, so that is what I am trying to say, that it develops memory as well. You will get to know about that, how it develops memory and all. Does it go to the brain and say that please you have to remember this? No, it does not work like that. There are certain other things. Let's see. So whenever we get a disease, okay, whenever we get a disease, so probably if you might have seen people when they got COVID, the first time they got COVID, it was our body was not able to really fight with it. Okay, our body was not able to really fight with it, and it was a very low intensity response that was given by our body. Now this response is known as primary response. The first time when an antigen comes, okay, our body is not very strong. It does not know. It is like a new person getting inside our house, so we do not know how to behave. But with time, when we get comfortable with them, obviously we are going to show our true colors, right? Similarly, this pathogen, when it comes for the next time, our body will initiate next set of responses which are known as secondary response. Now this will be rapid, this will be intense, such that this particular pathogen will be completely killed most of the times. So first is primary response that comes in our body, and you have to remember it is a very low intensity response. Now our body will have the memory that okay, this pathogen has come, and next time if it tries to enter my body, I have to kill it. So next time when it comes, our body initiates the next response, that is secondary response, and it will be rapid and an intense response. Now who is behind all these responses? Who is responsible for this immunity? Let's see. So immune responses basically are of two types. One is humoral and the other one is cell mediated. Humoral as in, humor term means fluid. The term humor means fluid. So basically inside our body, inside our blood, we have so many cells, and these cells are going to fight against the diseases. Okay. Now cell mediated means cell itself is going to do something. I'll make you understand all of this thing really well. Just have some patience. Okay. So let's talk about humoral immunity first. So the one that plays a major role in humoral immunity is B lymphocyte. What is it? It is what? B lymphocyte. So here I have written B cells, that means it is what? B lymphocyte, and you know that it is a type of white blood cell. We just studied about it, right? Now this B lymphocyte, when it gets activated, we will get to see how it gets activated, but when it gets activated, it releases two types of cells. One is plasma cells and the other one is what? Memory B cells. Now plasma cells now will be giving rise to antibodies. I told B cells are the ones that is responsible for antibodies, right? So in B cells, when they get activated, they release two types of cell. One is plasma cell, other is memory B cells. Plasma cells are going to release antibodies. Memory B cells have a memory of it. Will have a memory of it that this particular antigen, okay, so this particular pathogen was there and next time it comes, I would have to kill it. So this is wanted there. Okay. Now in cell mediated immunity, the major role is played by T cells. So T lymphocytes plays a major role over here, and they also have different type of T lymphocytes. Okay, it's not like when they get activated, there are different types of T lymphocytes. Here when it gets activated, we're releasing these two cells. It's not like that. Here these are different types. We have T helper cells, which is going to help other cells. Which is going to help other cells. This is the helping friend. It can help either a B cell or it can help a T cell. T killer cells are directly going to kill the cells. And memory T cells are going to, again as I said, it is going to store the information that this was the person who tried to enter our home without our permission, and now for the next time when it comes, we have to kill it. Apart from that, there are T regulatory cells and lots of other cells. As of now, we don't have to learn about them. T killer cells are also known as cytotoxic cells. Okay, fine. I'll tell once more. Okay, when you spoke about acquired immunity, it is pathogen specific. For different pathogens, we have different type of things being released on our body. Second thing, whenever an infection happens in our body, there will be, whenever an immune response happens in our body, there will be memory of it. Okay, so the first time it comes, it is known as primary response. This will be low intensity, and the second time it comes, it is known as secondary response, and that will be rapid and intense. Now, who is responsible for this immune responses? Two things: either the response can be humoral response or it can be cell mediated response. Humoral response means here B cells are involved, and B cells, when they get activated, they release plasma cells. Plasma cells release antibodies, and or they can obviously, when they do this, they also produce memory cells so that they have a memory of what was the pathogen that caused this whole ruckus. Now, cell mediated immunity is mediated by T lymphocytes, and T lymphocytes, as you can see, there are different types: T helper, T killer, and memory cells. T helper is going to help. You will see how it helps. T killer cells are cytotoxic cells. They are going to kill the other cells by releasing perforins, just like natural killer cells. Okay, and memory T cells stores the information. So, let's talk about humoral response first, and for understanding humoral response, you have to understand the structure of antibodies. So, in your textbook also you have structure of antibodies given, right? So, you might be seeing that there is something like this, then there is something shorter. So, this long thing that you see, it is known as, heavy chain. Obviously, when it is long, it has to be heavy, right? And the small thing that you see over here, it is known as light chain. Okay, smaller, so it will be lighter as well. Now, what is the thing that you have to remember? These these heavy chains, they are connected to each other with the help of disulfide bonds. So, there will be two sulfide. There will be bonds, and this bonds, how many disulfide bonds will be there? I am writing over here, disulfide bonds. Now, how many of them could you find over here in the in the heavy chain? 1 to 13. 1 to 13 disulfide bonds you could find in between the heavy chain. Now, the heavy chain thinks that okay, I have to connect to light chain as well, or it might feel alone, right? So, over here also you had disulfide chain, but this is only one. So, here also you have one, here also you have one. It's not 1 to 13 just like how you had over here. Okay, so we have a heavy chain, we have a light chain. Now, children, all these things that you see over here, they are made up of proteins. They are all polypeptides. Okay, what do you understand by polypeptides? Basically, proteins and everything, they are made up of amino acids. So, amino acids basically join together and form a chain like this. Similarly, amino acids join together and form a light chain like this. So, all of them, they are made up of proteins, and what do you call them? Immunoglobulins. Will you be able to remember it? Immunoglobulins. Okay, These antibodies are known as immunoglobulins. So in our blood we see that there are globulin proteins. What are these globulin proteins? Basically the antibodies itself. So these antibodies, some of them are freely floating in a secretory phase in the blood. So those are the ones. Now talking about one more thing. So I said heavy chain is there, light chain is there, and they have attachment 1 to 13 disulfide bonds, or here 1 disulfide bond. Now basically over here this antibodies at this particular position that you have seen, it is known as, okay, I should write it properly. This particular region that you see over here, I should erase the wait, where is that? Yeah. You see these sites, if I zoom this in, then I think it will be more clear to you. There are sites over here, okay? There are sites over here as well. What do you call these sites? You call the sites as paratope. You call the sites as paratope. Okay. Now, what is the use of paratope? This is the place where an antigen, for example, an antigen comes and attaches. Okay. And this will ignite a whole set of reactions. Well, now, antigen will also have a site for attaching to the paratope. What do you call this site as? Epitope. You say epitome, right? Epitome of success is something that is highest. So, similarly, epitome is the highest region over here, where epitome is the part of the antigen itself, which is going to attach on the paratope. You can remember E comes first before P. So, E is the one that comes and attaches over P, that is paratope. Okay, I hope that is also clear. So this is all about our antibodies. Now we have different types of antibodies as well, children. Where can I make that? Oh my god, I would have fallen now. Okay, so basically I have immunoglobulin A, or I shall start with a trick. So remember this always, gamed. Will you be able to remember this? Gamed, okay? Where there is IgG. IgG is the one, is the only antibody that is transferred from the placenta to the embryo during the growth. Okay, it is the first antibody to get transferred. So it comes from the placenta. You can remember like that. Talking about IgA. IgA. So basically again this is also transferred from the mother to the baby during lactation. So you might have seen that people should be, mothers should be breastfeeding their children during the initial days, because they have something which is known as colostrum, the first milk, yellowish milk, which is rich in antibodies. So babies, they cannot make their antibodies as of now. We have to transfer them for some time at least. So basically IgA is being transferred. So this comes from the milk. Initially this is transferred from the milk. Now talking about others, we have IgMU. Talking about others, we have IgM and IgD. Both of them, this and this, both of them are present on B cells. On the top of B cells, you will be finding, IgM and IgD. So they are going to help in response. Okay, so that is there on your B cells. Now talking about IgE. IgE is responsible for, very much important, it is responsible for allergy. Remember IgE, allergy, it sounds similar, and also remember eosinophils, it also started with E. They were also responsible for causing allergy. So all the allergic responses are caused with the help of IgE. You will get to know them also. So we spoke about antibodies. Antibodies come as a part of humoral response, because B cells are the one that releases plasma cells, and these plasma cells are going to release antibodies. We have 5 of them gamed. Okay, one is G. Okay, the first one is G. G is passed on from the placenta itself, IgA from the milk, IgM and IgD on the B cells, they are present, and also they are found in the secretory mode, that is in the blood, but it is comparatively lesser. The most common thing that you will finding in fluids will be IgA. Okay, now IgE is responsible for causing allergic responses. Now let's see the working of humoral response. So basically, there's a B cell. There's the first condition that I'm going to tell, okay? There's a B cell. This B cell saw an antigen. This B cell saw an antigen, or like, oh, now basically if the antigen comes and attaches on the antibodies, it can, this particular B cell itself can release what? Plasma cell. Sorry, it can produce plasma cell and what? Memory cells. If it releases plasma cell, then obviously antibodies will be released. Now when antibodies are going to get released, obviously it can act on the antigen. It can call more cells to protect, to what? Avail the protection. That is one thing. Okay, either you call more if you have more antibodies, that means more cells will be coming along, more macrophages will be coming along, and then they will be phagocytosing the organism and killing it. Okay, that is one thing. Or memory, because obviously you will be having a memory of what was the one that came and attached and causes the whole problem. Now second situation is when the macrophage is there, that is your phagocytosis cell, right? You remember they were doing phagocytosis. Now they have a very important protein at their top, okay, which is known as major histocompatibility complex, MHC. How can I make it? Okay, I'll make one over here, MHC2. What is it known as? Major histocompatibility complex. Okay, so basically macrophage took in the pathogen, killed it, and now there are small peptides of it. When you break these things down, you will get small peptides, and there should be one protein, one complex that should be showing that, okay, we have certain peptides, that means this particular cell was responsible for killing a pathogen. Now all the other cells would be made aware about the entry of a pathogen and that it was killed by the macrophage. I'll tell you once more. For example, when we fight, okay, when we fight for a country or things like that, we put a flag there saying that this is our property. Similarly, or showing, or probably you are just stuck somewhere and you want to wave, you want to show that I want help. What do? You wave some flag off, right? Similarly over here, the macrophages, they took in the antigen, or they took in the pathogen, they killed it, and now they put certain portions of this pathogen outside with the help of MHC2. Okay, there are MHC1, there are MHC2. Over here I'm talking about MHC2. Now this MHC2 is presenting the pathogen. Together, whole cell, you call it antigen presenting cells. Okay, as of now, just forget whatever I just said. Just imagine that this is there and it is being shown. Now TH cell, that is T helper cell, sees it. Okay, there is something, I have to go there. T helper cell sees it, comes over here, and when T helper cell comes and attaches over here to the macrophage instead. So this is the first thing that they did. Second thing is that this happened, and the third thing that happens is the macrophages will be releasing interleukins. Which now will be activating the TH cell. Okay, so TH cell has just come attached. That's the only thing that happened. It's not yet activated. For activating it, when this attachment happens, the macrophage will be releasing something that is known as interleukins. So interleukin 1 is being released. Interleukin 1 is being released. Okay, interleukin 1 is being released and it acts on the TH cell. Now it has become active and they will again release so many other cells. For example, B cell stimulatory factor or macrophage stimulating factor. In this way, it will call all the B cells to come over here. There is some problem over here, you have to come over here. Now B cells will obviously come. Okay, so basically what happened, interleukin act, TH cells are now active, TH cell will be releasing certain proteins, they will be releasing certain proteins, because of which the B cells will be coming at this particular site. Okay, so release of proteins will be the fifth point, and B cell coming to this particular site will be the 6th point. Now they will be seeing that something has happened, and B cells will now be releasing what? Antibodies. This is what? 7th point. Okay, confused? I can make it easy for you. There was a macrophage, it killed the pathogen, showed some part of it with the help of MHC2. Okay, now this MHC2 is recognized by TH cells. Only TH cells can only go and attach if there is MHC2. Okay, now they can go attach over here. It becomes activated. How it becomes activated? Basically, when it comes and attach over here, the macrophage release interleukin 1. Interleukin 1 goes and acts on TH cells. Now the TH cells will be releasing certain proteins, certain factors. Okay, certain factors. I should be writing certain factors. And now these factors will be calling upon B cells. And these factors will be helping for the accumulation of B cells as well as more macrophages. The B cells, if it comes, it can go and say that, okay, something has come and this is the antigen. We have to produce what? Certain antibodies specific to it, and the antibodies will be produced, and the memory cells will be also produced. Okay, so before producing antibody, obviously you need plasma cells. Plasma cells is the one that produce antibodies. This is how humoral response work. Over here you see a major role of B cells, and TH cells have helped them. Usually this is the pathway that is being followed, but sometimes B cells alone can also go and find some antigens and okay, so basically you have to understand one thing. There is only one macrophage that has done it. You need to call more macrophages to kill the whole amount of pathogen that is inside the body, which is why the whole drama is happening. Okay, if there is no force that is there for killing, obviously nothing could work. So we need more macrophages, we need more antibodies and everything. Similarly over here also, basically this is enough, but you know, during severe infections, we need a very huge and a big army to fight against the disease. So this is the whole thing. Okay, so that is how humoral response works. We'll read it out once and then we'll go ahead. So acquired immunity, on the other hand, is pathogen specific. As I said already, it is characterized by memory. Okay, this means when a body encounters a pathogen for the first time, it produces a response known as primary response, which is of low intensity. Subsequent encounter with the same pathogen elicits a highly intensified secondary, or you also call it anamnestic. Okay, that also means the same thing, that is second response, anamnestic. Now this is ascribed to the fact that our body appears to have memory of the first encounter, which is why it is doing a secondary response, right? Now the primary and secondary immune response are carried out with the help of two special types of lymphocytes present in our body, that is B lymphocytes and T lymphocytes. So in both primary and secondary you have B and T lymphocytes both. You saw the help, right, how T helper cell was helping. Now B lymphocytes produce an army of proteins in response to pathogens into a body to fight with them. What are these army of proteins? Immunoglobulins. Immunoglobulins, that is antibodies. These proteins are called antibodies. The T cells themselves do not secrete antibodies but help B cells to produce them. So why did I explain all this thing? Because I want to explain this. You didn't know. If you do not know, if you just read the NCERT, obviously you won't get the hang of it, which is why I explained. Okay, each antibody molecule has 4 peptide chains, two which are known as light chain and 2 longer known as heavy chain. Hence the antibody is represented H2L2 because we have 2 heavy chains and 2 light chains. Okay, and each of them, they are made up of peptide. They are peptide chains. Peptide chains, as in when amino acids they come together, you call this as peptides. Smaller amino acids coming together, as in smaller chains when they are formed, you call it peptides instead of protein chain. Same thing. Okay, so amino acids are the one that makes peptides. Peptides altogether are the ones that makes bigger proteins. Okay, so since they are small ones, we call it peptide chain itself. Okay, different type of antibodies are produced in our body. IgA, IgM, E and G are some of them. These not mentioned here. Okay, a cartoon of an antibody is given in your book. Because these antibodies are found in the blood, the response is known as humoral immune response. These antibodies, since they are present in the blood, which is why this is known as humoral immune response. This is one of the 2 types of our acquired immune response, antibody mediated. Okay, now the second type is known as cell mediated immune response or cell mediated immunity. cell mediated immune response and cell mediated immunity. The T lymphocytes mediate CMI. Let's read it and then we'll go ahead. Or they have written anything? No. Chalo, then we should go ahead. So basically, this is all about cell mediated response. Okay, you listen to me very carefully. It's very easy to understand. Now this macrophage, as I said, similarly had an encounter with a pathogen. Okay, killed it, and now it is presenting it over it. So over here, this particular molecule that helps in the presentation of the antigens, what do you call? Just imagine, this is MHC2, major histocompatibility complex 2. So every one of us has different major histocompatibility complex that is made up of different things. Proteins itself, but in a different order. Just like how we have different fingerprints in our body, we have our body and some other person's body will be having MHC in a different way. Okay, different types. So basically, yes, TH cells, they see it. Okay, they see it, and now when they go there, they become now macrophage will be releasing something that is known as interleukin-1. Interleukin-1 will be activating the T cells. TH cells will be now releasing macrophage activating factor. That means it will be calling upon more macrophages. It will be calling upon more macrophages, or it can also call it can also release B cell stimulating factor. B cell stimulating factor. In this case, what will come, children? B cells will come, and B cells, as you know, they will initiate with the help of antibodies. They will release plasma cells and then antibodies. So what I was saying is that there is a macrophage. It is killing the pathogen and it is presenting it over, so that there are other pathogens also. You need kill, you need force. So you're just calling other cells to come to this particular site and help them. So MHC2 is the one with the help of its presence. Now the TH cells, T helper cells. Okay, this is what? T helper cells. They see it, they go there. Now when it comes in contact, it becomes activated. How it becomes activated? Because macrophage will be releasing interleukin-1. Interleukin-1 goes and acts on TH cell. Now the TH cell will be releasing macrophage activating factor, calling around more macrophages at the site of infection. Next thing, B cell stimulating factor could also be released, calling B cells for the presence of antibodies. Now there is some other situation. Okay, here both of the cells, you can see that it is cell mediated. Cell is the one that is doing everything. All the responses are being done by a cell predominantly. Okay, in there when you talk about humoral response, antibodies were the one that had the major role, even though cells were involved there, but antibodies release is the major thing there, and antibodies are released in the blood, which is why it is known as humoral immune response. Now over here, macrophage again encounters something, presents it. Okay, but this time here, instead of MHC2, TC cells, or TC, that is cytotoxic cells or killer cells. Okay, basically they understand that there is a problem only when MHC1 presents the peptides. Okay, MHC1, when they are presenting the antigens, that's only when TC cell can understand. So their language and their language is kind of different. So here it was MHC2 who was presenting, here it is MHC1. Now TC cell or cytotoxic cells or killer cells, okay, same thing, interleukins, they get activated. TC cell will be now releasing perforin and cytolysin. Perforin, as you know, it causes perforation of whatever cell is the direct killing is there. You don't need to call upon macrophages on all of them, direct killing. Okay, perforations is being caused, and because of this perforation made in the body of the organism which is causing the disease, obviously it will die, or it releases cytolysin. Cytolysin, as I said, it will kill the whole cell. It will release certain factors which is going to kill the whole cell as a whole. Okay, and they call upon macrophages as well. There are other factors being released to call upon macrophages if the whatever thing is there, it is very intense. But basically they do their own function very efficiently. Okay, now that is about cell mediated immunity. Here T cell is acting as a major thing. Now when you say that, okay, for example, in my body some organ failed and I want another person to help me by giving an organ, donating an organ. Okay, for example, if it is a person, a friend of mine, okay, a friend of mine who have given me, there are higher chances that this particular organ will be rejected. This particular graft, probably a skin portion or anything that is there, it could be rejected. Why does this rejection occur? Why does this rejection occur? Because our body thinks that this is foreign, and if something is foreign, we have to kill it. Okay, and this graft rejection happens because of cell mediated immunity, that means T lymphocytes. So as I said, different people in their cells, they have different MHC. The MHC that is being made, it is different in all of us. So basically just like fingerprint. So our body senses this and creates a response, that is cell mediated response. Please, it is very, very important in graft rejection. Who has a major role? It is cell mediated immune response. And here T lymphocytes are the one whom we should blame. Now there are two types of graft. Okay, autograft is there. I'll write it here itself. There is autograft where from my body only, for example, I have a skin over here, I want to put it somewhere else because that somewhere else is wounded and I need attention over there. So basically that is being sealed, and over here that has been put, that is autograft. Okay, now there is something that is known as allograft, where something that is non-self, it's not a part of my body, is being put. And for doing this grafting, there is so many techniques. You have to see if the tissue is matching, if you have to see if the blood is matching, lots and lots of things are there, which is why not all the grafting techniques are, let's say, a huge success. Most of them are a failure because of the same reason, because our body could react to it differently, because they do not know that it is something foreign, but we are still trying to help. So that is one thing. Okay, so that is about graft rejection. We'll just have a quick read on that and then we'll go ahead. Very often when some human organs like heart, eye, liver, kidney fail to function satisfactorily, transplantation is the only remedy to enable the patient to live a normal life. Then a search begins to find a suitable donor. Why is it that organs cannot be taken from just anybody? What is that the doctors check? Grafts from just any random source, an animal, another primate or any other human beings cannot be made, since the grafts would be rejected sooner or later. Tissue matching, blood group matching are essential before undertaking any graft transplant, and even after this the patient has to take immunosuppressants. Why do they take immunosuppressants, children? So that our body, we are telling our body, please do not show immune response. We do not want immune response. If they are showing immune response, obviously what will happen, they will identify this is foreign, you have to kill this, which is why immunosuppressants are taken to suppress the immune response. Okay, all their life. That is why they are prone to other infections as well. Okay, the body is able to differentiate self and non-self, and the cell-mediated immune response is responsible for graft rejection. Very, very, very important for NEET. Okay, and I hope you got this. Now, grafted kidney may be rejected in a patient due to, tell me, I just taught you right now. Grafted kidney may be rejected in a patient due to what, children? Now you know cell mediated immune response. You tell me, if you have not studied any of this thing, would you be able to answer this question? That is why I am saying it is very much important to attend the classes as well, to understand certain things if at all needed. Okay, so cell mediated immune response is the correct answer. Going ahead with active and passive immunity, this is my favorite topic, and this is the portion that has given so many number of questions. Okay, so let's see. When a host is exposed to antigens as in any pathogen, okay, which may be in the form of living or dead microbes or other proteins, or other proteins. Look, listen, what is the scenario over here? Host, as in for example me. I am exposed to some antigens. This antigens would obviously ignite some immune response in me. Okay. Now this antigen can be in the form of living. It can be a living antigen, or it can be dead also. Okay. Now when they come inside my body will be releasing what? Antibodies. Antibodies are produced in the host body. This type of immunity is called active immunity. So without me doing anything, I am not doing anything. Antigen is trying to get inside my body itself is producing antibodies. This is active immunity. My body is really actively producing something. Okay. Now active immunity is slow, because obviously my body is seeing it for the first time. It does not know it, so it will be slow, just like how the primary response, right? And takes time to give its full effective response. So slow and time taking. Injecting the microbes deliberately during immunization or infectious organisms gaining access into the body during natural infection induce active immunity. So active immunity can be induced in two ways. Okay, either one infection comes, I get affected because of the infection and my body produces something, antibodies against it, or basically to make my body produce antibodies, what I am doing, I am injecting the microbes deliberately. It's not like I am injecting a living microbe, it can be dead. In vaccination it happens, right? So basically I am injecting microbes in my body so that my body could produce antibodies now and also the next time if the infection comes. This is also known as active immunity. Did not understand? I'll tell you. So for active immunity, always remember one thing. Our body, in active immunity, our body is going to release antibodies. Okay, it will be our body. Now natural is when you have some infection, some disease or any natural infection. Obviously, to fight against this, my body will be releasing antibodies. Agreed? This is happening naturally. Or else what do? Artificial is when I am putting in the disease causing organism over here. Okay, I am putting the disease causing organism artificially. I do not have any disease, but I am putting it over here. Okay, such that my body produces antibodies. Now this is on this principle vaccination works. Vaccination works on this principle. You will get to know about that. So active is two types, natural and artificial active immunity. Okay, so inactive, how you can remember, our body, whatever you do, our body is going to release the antibodies. Either it can be a natural infection or you are giving some microbes, which is why they are producing antibodies, but it is obviously our body. You have to remember it. Okay, this is why people get confused. That is why I am telling it 10,000 times. Now passive immunity, when ready-made antibodies are directly given to protect the body against foreign agents, it is known as passive immunity. When ready-made antibodies are given. Okay, so natural when our body itself, so natural when our body, okay, over here passive means you are getting antibodies, you are not making, okay, I'll write it, you are not making antibodies, you are getting antibodies. From where? From where can ma'am can we why antibodies? No, it's not like that. Natural as in you know from mother to child, basically one thing, from the placenta I told you. What is the first antibody, children? Gamed G, IgG is being got. Then from the milk, the first milk, which is also known as the yellowish milk, which is known as colostrum, they give IgA. Remember, okay, gamed, next letter is A, right? So all of them, you are giving ready-made antibodies over here, and this is occurring naturally, nowhere else you can see this. Now there is something that is known as passive immunization, where I am giving ready-made antibodies. For example, a snake. Okay, there's a snake that come and bit me. Now I won't say, hey, my body, please produce antibodies. I cannot wait for active immunity to take place, because active immunity is definitely slow. I need antibodies as of now. So we do passive immunization. What is this known as? Immunization. You might be studying about this in next thing. We studied about vaccination also here and immunization also here, but it's okay. We'll re-study about it there also. So it is known as passive immunization, because you, for example, snake bite has come. You are going to put anti-venom, which is basically antitoxin or anti-venom, which is basically a mix of antibodies and all the other things which is required for you to survive there. Okay, or you have some, for example, tetanus. In tetanus also you put antitoxin, So antitoxin is again a mix of antibodies and everything. Mix of ready-made antibodies. And everything that is ready-made has to be fast. Its effect will be fast because your body does not need the time to make the antibodies. Antibodies, once it comes, it is going to be fast. But over here, your body is not making it, you are getting it, so which is why it is known as fast. And passive immunization means the same thing. Okay, it is very artificial. These both things are artificial, right? Over here, we are artificially injecting antigens for our body to produce antibodies. That is why it is artificial. Over here, we are giving antibodies itself directly. You are not letting your body produce antibodies also, you are directly giving antibodies, and this is because you got a snake bite or you have a tetanus infection, a lot of things like that. So this is what you mean by active and passive immunity. Again, I am telling you, in active, our body produces antibodies. It can be a natural infection or it can be because we gave something so that our body could produce antibodies at first. Next time if some infection comes, we know about it and we can produce antibodies at that point itself, and it would be faster. The secondary immune response would be faster then, which is why we do this. When vaccination, the exact same thing happens. In vaccination, polio vaccine and all, we are giving the attenuated form. What do you mean by attenuated form? I will tell that also. We say live attenuated form or dead. Attenuated means you are bringing certain changes in the organism. It's not fully dead, but it is not able to cause. It won't be causing it won't be causing any infection, but it can cause immune response. It has all the factors to cause the immune response, but it won't be causing a trouble. That factor of it which was responsible for creating a ruckus inside the body, it's being removed. That is being hampered. Just that particular thing which is responsible for creating an immune response, that is left back. That is what you mean by live attenuated. You are bringing certain changes. You are making the pathogen weak. Okay, there is the meaning of attenuated. Or you can put dead, dead microbes inside your body for producing antibodies. That's the exact thing that happens in vaccination. So you know, in COVID also we got vaccine. What did this vaccine contain? That this particular vaccine was containing spike proteins. So in the COVID virus, so over here there were spike proteins which were responsible for creating the immune response. They cannot cause a trouble. They cannot cause an infection, but they can give an immune response because saying that okay, this is something foreign that has got inside our body. So they will be obviously producing antibodies such that when next time COVID comes and attacks us, at least our body will be prepared for it. Okay, so we have studied about active and passive immunity, and also we spoke about vaccination and immunization here itself. But still we will read it once more so that you get an idea of it. Okay, please remember natural placenta, IgG, milk, IgA. Now, when ready-made antibodies are directly given to protect the body against foreign agent, this is known as passive immunity. Do you know why mother's milk is considered very essential for the newborn infant? You know that, right? So yellowish fluid, colostrum secreted by the mother during the initial days of lactation has abundant antibodies, that is IgA, to protect the infant. The fetus also receives some antibodies from their mother through placenta during pregnancy, and what is this? This is IgG. Okay, so over here they have mentioned about natural active immunity, artificial active immunity as well. Okay, when they are saying that injecting the microbes deliberately, that is artificial. When they are saying that infectious organism gaining access to our body, that is natural. Over here they have mentioned about natural passive immunity, where we are giving the antibodies from the mother itself. Now we are going to talk about artificial. So before that we have a question to solve. Tell me the correct answer for this question, children. Colostrum, the yellowish fluid secreted by mother during the initial days of lactation, is very essential to impart immunity to the newborn infants because it contains what? What is there in colostrum? Tell me. Immunoglobulins A. You do not know immunoglobulin, that is a trouble. IgA, immunoglobulin A. Okay, came in the year 2019. Short-lived immunity acquired from mother to fetus across placenta or through mother's milk to infant is categorized as. Tell me, is categorized as. It is short-lived. Yeah, with this I want to tell you one more thing. What is that thing that we acquire from mother across the placenta and mother's milk? It is passive immunity. But with this, I want to tell you one more thing. Going back to active and passive immunity, this might be slow. This might be slow, but it has long-lasting effects, because once antibodies are produced, it's all good. Memory cells and everything is there. But when you're injecting antibodies, can it be long-lived? No, which is why we are requested to take shots. Okay? This will be short-lived. Until the number of antibodies and everything is done, we are okay. After that, not okay. Okay? Okay, let's talk about vaccination. The principle of immunization or vaccination is based on the property of memory of the immune system. In vaccination, a preparation of antigenic proteins of the pathogen or inactivated weakened pathogen are introduced into the body. In vaccination, this is happening. What are we doing? Antigenic proteins, that is foreign proteins of pathogen or inactivated weakened pathogen is put. The antibodies produced in the body against these antigens would neutralize the pathogenic agents during actual infection. Now, the antibodies which will be produced in the body against these antigens will obviously neutralize the effects of the pathogen during actual infection. When the infection actually comes, that is in the second time, it will show all its effect. The vaccines also generate memory B and T cells. The vaccine, it generates what? Memory B and T cell that recognize the pathogen quickly on subsequent exposure and overwhelm the invaders with the massive production of antibodies, because both of them, they have memory cells. Okay, so vaccination, what does it induce? Both B and T cells. Obviously, your body is producing antibodies and T cells, lymphocytes, everything is getting activated. If a person is infected with some deadly microbes to which quick immune response is required, as in tetanus, we need to directly inject preformed antibodies that is already there. You got it from your body itself or someone else's, but you already have the preformed antibodies. Or antitoxin. What is antitoxin? A preparation containing antibodies to the toxin. That is what you are talking. What do you mean by antitoxin? So you already have it. Now you just have to inject that. Even in the case of snake bites, the injection which is given to the patients contain preformed antibodies against the snake venom. So whatever snake venom is there, against it whatever antibodies are required, that is being injected to the human body. Now this is known as passive immunization. What do you call it? Passive immunization. Over here you are handing over the antibodies directly, which is why it is known as passive immunization. Even though they have not written this under active immunity category, but still I am going to tell you, this is a principle. Active immunity is the principle that on the basis of which this vaccination works. Okay. Now, apart from both of them, recombinant DNA technology has allowed the production of antigenic polypeptides of pathogen in bacteria or yeast. What it is saying, recombinant DNA technology, as in when you have a DNA and you are altering the DNA for your own purpose, that is what you know as recombinant. You are mixing and you are doing some changes in your DNA. Okay. It has allowed the production of antigenic polypeptides of pathogen in bacteria or yeast. Vaccine produced during this approach allow large scale production. So whatever vaccines are being produced, it is being as in they are making what say certain antigenic things, so that could ignite a immune response in our body. Okay, so this is being done to make the vaccines. What is vaccine? Basically the preparation of antigenic proteins of pathogen or basically inactivated or weakened pathogen, anything you are making with the help of recombinant DNA technology, so that we have enough amount of vaccine. So we cannot obviously wait for the infectious agent to come and then infect us and then we make vaccine out of it. No. Okay, we have this technology, recombinant DNA technology, according with the help of which we already make the vaccine, and this vaccine is given to all. Okay. And hence, okay, what does it do? It gives large scale production and also greater availability for immunization. Example, hepatitis B vaccine produced from yeast. So hepatitis B vaccine was produced from yeast. Give her 5-star. You never know what questions could come. Hepatitis B vaccine is produced from yeast, and also vaccines generate B and T memory cells. Okay, done. Can we go ahead? Okay. The story of vaccination I already told about it, so I am not mentioning it again. Talking about allergies. Now that is something very interesting. When you have gone to a new place and suddenly you started sneezing, wheezing for no explained reason, and when you went away your symptoms disappeared. This has happened to you. Some of us are sensitive to some particles in our environment. The above mentioned reaction could be because of allergy to pollens, mites, etc., which are different in different places. The exaggerated, now this is the important thing, okay? The exaggerated, as an over, okay, response of the immune system to certain antigens present in the environment is known as allergy. So this is not a normal case. This is exaggerated immune response. We do not want this much exaggerated immune response. The substance to which an immune response is produced are known as allergens. So something that causes allergy to me is known as allergen. The antibodies produced to these are IgE type. I told you IgE are the one that is responsible for creating allergic responses. Common example of allergens are mites, induts, pollens, animal dander, etc. So even now, when you see me, my nose is also red, if you are able to see. Why? Because I am also allergic to something that is there in this particular studio. Okay, and this is because my IgE is very active. Now if it goes very active, then probably I will be sick, and that usually happens as well. So that is there. Now symptoms of allergic reaction include sneezing, watery eyes, running nose, and difficulty in breathing. Why does all of this happen? I'll tell you. Allergy is due to release of chemicals like histamine and serotonin. So histamine and serotonin are released from the mast cells. So there is some cell like mast cells, and you do not know anything about it. So let's talk about it. So again, the story begins, same thing, okay? Macrophages, the one that causes allergies there inside. It is being shown with the help of MHC2. Now this comes and binds, this becomes active, interleukin. Now until now you might have, it might be pakka feeded in your brain, okay? Now TH cells will be releasing certain factors, B cells stimulating factors. So that B cells will be coming, B cells release plasma cells, memory cells, plasma cells releases antibodies, that is IgE mainly. This IgE will now go and bind to the mast cells, okay? These are the mast cells, and mast cells, children, are present everywhere around the mucosal lining and things like that, in our throat region, in a gastrointestinal region, lots and lots of places. Mast cell is responsible for allergic responses, okay? So IgE is there, it binds over it. Now, the pathogen already informed by the macrophages, right? Now the pathogen will come and attach over it. Mast cell will quickly release histamine and also serotonin. Serotonin, yes, serotonin. Now what is the role of histamine and serotonin? Basically, histamine is responsible for vasodilation. Vasodilation. That means your blood vessels, which are like this, as I told you, will be inflamed, and now the vessels would be like this, so that all the cells which are required at this particular site of infection can come. All the mast cells and everything could come and give out their secretion, kill the pathogen which is causing a problem there. Now this is a very exaggerated response, because sometimes this, but obviously we do not need this big response for something that might be solved very easily, right? So this is that part of our body which is responsible for exaggerated response, and this is why we call it an allergy. Okay, and who is responsible for it? IgE is responsible for it. IgE goes to mast cells. Mast cells are the major cells involved in allergic responses, because mast cells are the ones that releases histamine and serotonin. You remember it? Histamine and serotonin. Histamine, as you can see over here, H also looks like this, okay? So basically it is vasodilating the blood vessel so that all the cells could reach at this point and say that this is some trouble and you have to solve it, okay? If the pathway is right, can everything reach over here? No. Okay. So that is also there, and apart from it, it causes inflammation, lots and lots of things, which is why. And also, what do we have? The mucus and everything. The mucous lining becomes very thickened. We will be having sneezing, we will be having watery eyes, because the system in serotonin, everything is going to affect all these regions of our body. The mucous lining will be thick, which is why we have coughing, we have sneezing and things like that, because mast cells, they are very efficiently present over here and there in the nose cells. Lots of things are there. So I hope you got the basic concept of how allergy is caused. Okay. Now, for determining the cause of allergy, what do we do? How do we understand that we are allergic to this? Basically, the patient is exposed to or injected with very small doses of possible allergens. Whatever, for example, I have eaten something, but I don't know what is in the whole day. At the end of the day, I got allergy, but I don't know what is that something that has caused me allergy. When we go to doctor, they give out some injections or they put something in a very limited form, not in a form that would kill me, obviously. Okay. Now, the use of drugs like antihistamine, adrenaline and steroids quickly reduce the symptoms of allergy. Antihistamine, because we do not want that much histamine to be released, that is causing vasodilation and all this trouble, making my mucous lining thicken, nothing. I don't want that, which is why we use antihistamine. You might have seen people who are very allergic and they have cold very frequently, they take cetirizine, because it is also an antihistamine. Okay, it is making the amount of histamine lower down in our body. Now, somehow modern day lifestyle has resulted in lowering of immunity and more sensitivity to allergens. More and more children in metro cities of India suffer from allergies and asthma due to sensitivity to the environment, because probably our immune system is very weak and does not know to differentiate between what it has to do and what it does not have to do. Okay. While I say this, I think I'm getting an allergy again. So this could be because of the protected environment provided early in life. So you should not be providing a very protected environment for your kids. They should be also playing around things so that they have an exposure to antigens early in their life and they could produce antibodies, and that antibodies could give out a good secondary immune response later on. So yes. Now we come to autoimmunity. As you know, the term autoimmunity means our immune cells are messing up, children. They are messing up big time. Why did I say that? Basically what happens, our cells usually have this thing. Okay, this is our cell. Usually it understands that, oh, this particular cell which has come, it is from a different body, but this particular cell is from the same body. That means it has the ability, we are the complex organisms, through evolutionary mechanism we were able to understand that, okay, this is foreign and this is ours. But with time, due to some genetic factors, due to some effects, we do not know why, our cells, some of them sometimes have lost this ability to recognize what is self and non-self, and then kills both. But when you're killing your own cell, that means you're destroying some part of your body, right? This is what you mean by autoimmunity. Okay, so we have autoimmune diseases where we are killing our own cells. The cell is not, our immune cells are not able to differentiate between self and non-self. No difference. Okay, can you give an example of this, ma'am? You know rheumatoid arthritis. Have you heard about rheumatoid arthritis? What happens in rheumatoid arthritis? Basically, this is how our things look. So you have a synovial membrane, children. Okay, you have a synovial membrane in the joints, and also you have cartilage. So this thing over here, they have mentioned that this is the synovial membrane which is there. So basically what happens, the synovial membrane, the cells of the synovial membrane are damaged. Our body is not able to recognize that, oh, it is our cell. But it goes and damages the synovial membrane. In a way, what happens, it will become swollen. Now obviously when you're doing something to our cells, they will also show certain reactions. They will get swollen. Now when they get swollen, what will happen, the cartilage which is there, because of this pressure and lot of things, the cartilage will now be somewhat broken. There would be some problem in the cartilage as well. Okay, the ligament that is over there. So basically what is happening, all the problems arise over here due to inflammation of the synovial fluid, which is caused because the synovial cells, the cells which makes up the membrane, are being damaged in some or the other way. Yeah, now this will wear our cartilage. Okay, it will make our joints very rough. Basically they would be sliding over each other. It will be causing pain. You will have inflammation, that's why you will have pain. This is known as rheumatoid arthritis, okay, which is an autoimmune disease. Apart from that, have you heard about Hashimoto? Have you heard about Hashimoto? This is a disease where the thyroid, in the thyroid gland, the thyroid cells are affected, and if the thyroid cells are affected, basically you won't be able to produce thyroxine hormone, and if thyroxine is not produced, then metabolic activities of bodies will be slowing down. There will be lots and lots of trouble. But eventually, obviously, thyroxine is synthesized, given in the synthetic format to people who have thyroid issues. Okay, so basically rheumatoid arthritis, Hashimoto, all of them are autoimmune diseases where our body fails to understand what is self and what is non-self and starts killing our own cells. Okay, so over here synovial membrane, the cells of the synovial membrane is affected. Okay, let's have a read of it as well. Memory based acquired immunity evolved in higher vertebrates based on ability to differentiate foreign organism from cells, that is autoimmunity. Okay, while we still do not understand the basis of this, two corollaries of this ability has to be understood. Why we have it? Okay, one, higher vertebrates can distinguish foreign molecules as well as foreign organisms, that is us. Most of the experimental immunology deals with this aspect. 2. Sometimes due to genetic and other unknown reasons, the body attacks self cells. So this is one thing that usually happens, but this is something that does not happen, but this has happened. This does not happen usually, but it has happened. This results in damage to the body and is known as autoimmune disease. Rheumatoid arthritis, which affects many people in our society, is an autoimmune disease which is caused by damage of cells of synovial membrane, which causes inflammation, which is why the cartilage becomes wear and then lots and lots of trouble happens during in the joint region. Okay, I hope that's also clear. Now, in higher vertebrates, chalo, do a question. In higher vertebrates, the immune system can distinguish cells and non-self. If this property is lost due to genetic abnormality and it attacks self cells, then it leads to what? What is it, children? Is it autoimmune disease? Don't write graft rejection over there. Okay. Graft rejection is where you know what is self and non-self. You're rejecting graft. This graft can be from outside source as well. This is autoimmune disease. Okay. Now, human immune system. With this, we are going to complete the topic. Let's do it really fast. Okay. The bone marrow is the main lymphoid organ where all the blood cells, including lymphocytes, are produced. So you know there is something that is known as bone marrow. Where do you see bone marrow and all? I'll tell you. So before talking about that, let's just start it. I think that's the best way. So basically, we have primary lymphoid organs in our body and we have secondary lymphoid organs in our body. Primary lymphoid organs are responsible for formation of lymphocytes, proliferation, maturation. Formation, proliferation and maturation is done in the primary lymphoid organs. What are the two primary lymphoid organs that we have, children? Bone marrow is one, thymus is the next one. Okay, so over here B, T cells, everything is being produced. But for them to perform their action, to perform their function of killing the cells or finding the antigens and things like that, basically they have to move to the other cells, other organs which are known as secondary lymphoid organs. And in the secondary lymphoid organs comes your spleen, your Peyer's patch, tonsils, and I'm forgetting one very important thing. We'll get to know that as well. So yes, lymph nodes. Okay, these are secondary lymphoid organs where what is happening? The lymphocytes basically come in contact with antigens and produce immune response. So this is your home and this is the place where you went to study. This is where you'll actually see what is life at the end. Okay, so basically that is the thing. Again, I'm telling you, primary lymphoid organs are the one where formation, proliferation, maturation of B and T lymphocytes and all the other cell happens. This occurs in bone marrow and thymus. These are the primary lymphoid organs. Secondary lymphoid organs are the one where the lymphocyte actually comes in contact with the antigens and now they start showing their function. Okay, so lymph nodes, spleen, Peyer's patch, tonsils are the one. We'll get to know about each of them. Here you have bone marrow. So basically just see inside the bone, no, the bone is not like, does not have anything. Okay, the bone has something that is known as bone marrow. There is red marrow, there is yellow marrow. Okay, initially our bone has more amount of red marrow and this red marrow has stem cells. You call them hematopoietic stem cells. Hematopoietic stem cells, which are responsible for formation of RBC, WBC and all, and platelets and everything. Okay, so bone have bone marrow. Which marrow? Red marrow. And this red marrow has stem cells. Stem cells, you know, they have an ability to divide into so many different type of cells. So they form the RBC, WBC and platelets. In WBC you have B cells and you have T cells, T lymphocyte, B lymphocyte, and all the lymphocytes that you studied, or the leukocytes that you have studied. Okay, but here our main focus is B and T. Now B lymphocytes matures here itself. It is formed here, it is matured here also. Okay, but when you talk about T lymphocytes goes to the thymus. Goes to the thymus, which is around there. Okay, and there it will undergo maturation, which is why it is known as T lymphocyte. T here stands for thymus. Okay, and where is the thymus located, children? Below the sternum. So there is chest bone or sternum, right? This is what? Sternum or chest bone. You might have seen that the ribs are attached to the sternum. Okay, this thing. So below it you will be finding thymus, where the T cells which are formed over here undergo maturation. So you know that bone marrow is there and your thymus is there. These are the primary lymphoid organs where formation and maturation also takes place. Later this red marrow, some of it will be turning into yellow marrow. Yellow marrow is responsible for the formation of fats and things like that. Okay, and giving rise to more bones. So that is the thing. You might have seen if you are a non-vegetarian, when you eat chicken legs, inside it there is a very solid thing, okay, that people eat. That is the red marrow, which is very much nutritious. Okay, now going ahead, these are the primary lymphoid organs. I hope that is done. Now just have a read of it and then we'll go ahead. The bone marrow is the main lymphoid organ where all the blood cells including lymphocytes are produced. So you know WBC, RBC, that is also being produced, and platelets also. The thymus is a lobed organ. Okay, it is a lobed organ, children, located near the heart and beneath the breastbone, that is the sternum. So it is located near the heart, beneath the breastbone. The thymus is quite large at the time of birth. So it keeps on reducing its size when we grow old. Okay, and reduces its size with age, and by the time puberty it is attained, it reduces to a very small size. Okay, so by puberty it has a very small size. Both bone marrow and thymus provide microenvironments for the development and maturation of T lymphocytes. So you know thymus is involved for T lymphocytes, whereas bone marrow is generally for B lymphocytes. Okay, thymus for its maturation, it has to go there as well. Okay, now spleen, talking about spleen and lymph nodes and stuff like that. Chalo, let's talk about lymph node first. What is lymph node? So basically I'll tell you one very important thing. You have your blood vessels, you have your blood capillaries. Okay, you have your blood capillaries which are very thin structures. Okay, now because they are thin and they are under pressure, okay, arteries gives its blood to the capillaries, and from the capillaries it goes to the veins. Okay, so while doing this, obviously oxygen is being transported to other cells which are surrounding it, and CO2 is being taken back. But since it has small pores, no, some amount of plasma which is there in the blood, and lot of things also move out. Plasma, lymphocytes, certain lymphocytes, and pathogens also. They also evade from the blood and gets into this particular space between the cells. Now there should be someone who could take these things back to the whole circulatory system, because we cannot waste our plasma or lymphocytes there. The blood pressure would be getting reduced, right? So this will open into a vein. Similarly, all this thing is collected and given back to a vein that goes to the heart. So what is that? Something that collects everything. It is known as lymphatic vessel, children. It is known as lymphatic vessel. And this lymphatic vessels, no, in between they will be having a lobed structure like this. And this lobed structure, no, it is very much rich in lymphocytes. They have so many lymphocytes. They have so many lymphocytes. So what were the secondary lymphoid organs? Basically the ones that will make the lymphocytes in contact with the antigens. And where are the antigens? Over here. So basically when all these things gets inside, When all this thing gets inside and you see there is a pathogen, this pathogen or this antigen is being caught by the lymphocyte which is there in the lymph nodes. So that is one thing, that is one secondary lymphoid organ. So over here you can find lymph nodes. Over here also we have lymph node, which is why doctor checks that whether it's swollen or not. When we have an infection or something, that might be swollen because they are fighting against diseases. Okay, so that is one thing. We have lymph node. Going ahead, we have spleen. So spleen is a structure that will be around our kidney, stomach. Okay, spleen is also known as the graveyard of RBC. Graveyard of RBC, because here the RBC, basically the old RBC, the worn out RBC are being killed, and their hemoglobin, all the proteins and everything, iron and everything is taken back for formation of new blood cells. But spleen is also an organ that is having enough amount of lymphocytes. So lymphocyte is there, okay, and RBC comes in, old RBC, worn out RBC. Oh, yeah, sorry, RBC I didn't make like that, I should not have made like that. So just imagine an old worn out RBC have come with a stick. This particular RBC will be killed by the lymphocyte itself in the spleen, or if not RBC, then any pathogen that comes inside again will be coming in contact with the lymphocyte and will be killed in the spleen, which is why I call spleen as a secondary lymphoid organ, because again it is coming in contact with the antigen over there, and we have enough amount of lymphocytes as well. Okay, this is known as graveyard of RBC as well. Now talking about the next thing, that is Peyer's patch, but before that I want you to read this. So let's read. The spleen is a large bean-shaped organ. It mainly contains lymphocytes and phagocytes. Phagocytes basically macrophages, which could take things and kill it. Yeah, it acts as a filter of the blood by trapping blood-borne microorganisms. Spleen also has a large reservoir of erythrocytes. Okay, it has a large reservoir of erythrocytes. The erythrocytes come there, gets killed, takes an hemoglobin, that is also there. And when we are small, when we are baby, at this point of time, spleen is also responsible for production of cells. That is something different. Okay, so it is known as a large reservoir of erythrocytes, basically because they're old and everyone, they come there for some time, they get killed later on, so which is why it's said like that. The lymph nodes are small solid structures located at different points along the lymphatic system. Lymph nodes serve to trap the microorganisms or other antigens which happen to get in the lymph and tissue fluid. Antigens trapped in the lymph nodes are responsible for activation of lymphocytes present there and cause the immune response. Okay, so they also cause the immune response. Now going ahead, there is a lymphoid tissue also located within the lining of the major tracts, respiratory, digestive, urogenital tracts, called mucosa associated lymphoid tissue mult, and it constitutes 50% of the lymphoid tissue. What does it do, children? It is constituting 50% of the lymphoid tissue. It is very important question, it has come before also. What is mult? I want to explain you that. So as I said, mult, mucosa associated lymphoid tissue. Okay, so basically the innermost layer, I told you there are 4 layers. If I have not told you, I am telling you now. There are 4 layers in our alimentary canal and everything. The outermost is serosa, then comes muscularis, the muscular layer, then comes submucosa, and then comes mucosa. So if you have one serosa, then muscularis, then submucosa, then mucosa. So this has to be the innermost layer, and it constitutes, okay, so mucosa associated, as in it is associated with this. So the lymphoid tissue which is associated with the innermost layer, that is mucosa, that comes under mult, and it constitutes what, children? 50%. Oh, not 60. I am saying 50 and it's coming as 60. It constitutes 50% of what? The lymphoid tissue. Okay. Now in this category comes 3 things. In this category comes Peyer's patch, tonsils, and appendix. Okay, so Peyer's patch is there in your small intestine, children. This is Peyer's patch and this is there in your small intestine. Peyer's patch will be rich in lymphocytes. Is it visible? Yes, it is rich in lymphocytes. So there are so many follicles of lymphocytes over here. So what happens, there are certain cells which are known as M cells. Okay, I'll write in black. M cells. These cells, they bring pathogen or antigens from the lumen of the small intestine, from the inside of the small intestine, that is known as lumen, okay, the empty space that is present through which the food and everything passes. The antigens are brought by the M cells to the Peyer's patch, and here they come in contact with the lymphocytes, which basically later on kill them. Okay, so Peyer's patch is one such lymphoid organ which is associated with the mucosa layer. Okay, which is associated with the mucosa layer. Actually, it is, so it starts with the mucosa layer, it ends in the submucosa layer, but it is there, it is associated with the mucosa layer. It has plenty of lymphocytes, and these lymphocytes, basically none of them go outside. Basically what happens, the M cells, there are certain cells known as M cell, microfold cells, okay, as far as I remember, it's microfold cells. They bring in the antigen, they come like, take, now you can react, and Peyer's patch is there in your small intestine. Okay, going ahead, we have appendix. So this was considered to be a vestigial part, something that is not useful, but it's not like that. It is useful. It is the place where good bacteria resides. Okay, and it is the first part that basically this is our large intestine, and appendix is over here near to our first part of our large intestine. Okay, and appendix is a place where there are so many lymphocytes again, and when antigens comes in contact with these lymphocytes, all of them dead. So people also remove the appendix, okay, thinking that's a vestigial organ, and when there's pain, obviously when there are grave situations, you should be removing it, but generally there's not a problem. Okay, so they are responsible for lymphocytes and also for harboring good bacteria. And then we have tonsils. So tonsils is not like just that we have it over here in the pharynx, in the throat region. We have different types of tonsils in the tongue, in the sideways. Lots and lots different types of tonsils are there. What is the use of tonsils? They are again a area where lots and lots of lymphocytes are there, and these lymphocytes are responsible for trapping the antigen. So when I'm having some food and there is an antigen, our tonsil traps it. And when the lymphocytes and everything attacks over that, that's when I say I have tonsillitis. Basically the infection increases way beyond. Okay, so that is about malt. So in malt comes all this thing, that is Peyer's patch, tonsils and appendix, because all of them, they are associated with the mucosa layer, and it constitutes 50%. Do not forget that. So that's all about the organs as well. Now let's solve this question. Some PYQ. Malt constitutes about -% of the lymphoid tissue in human body, came in 2017. Tell me the answer for this. I told you it's D. Okay, identify the wrong statement with the reference to immunity, children. So we have studied about passive and inactive immunity. You can tell me the the wrong statement regarding it. When exposed to antigen, living or dead, antibodies are produced in the host body. It is known as active immunity. It is correct, right? Antibodies are produced in the host body. In active immunity, it has to be our body that produces antibodies. When ready-made antibodies are directly given, it is known as passive immunity. Yes, it is known as passive immunity because our body is not making it, someone is giving it. Active immunity is quick and gives full response. Is it like that? Is it quick? It is not quick because it is not it cannot produce antibodies really fast. It is a very slow process because we have to rely on our body to release antibodies. So this is wrong. Fetus receives some antibodies from mother. It is an example of passive immunity. It is true. So this is the wrong statement. This came in NEET 2020. Very recent. So these were the questions that we did. Colostrum, the yellowish fluid, what does it contain, children? Immunoglobulin A. Interferons are synthesized in response to viruses. Short-lived immunity acquired from mother to fetus across placenta or through mother's milk to infant is categorized as what? It is short-lived, so has to be passive. Identify the wrong statement. We did it right now. In higher vertebrates, the immune system can distinguish cells and non-self. If this property is lost due to genetic abnormality, it attacks cell, then it leads to autoimmune disease. One question for you to solve, children. You can take a screenshot of this Antivenom injections contains preformed antibodies, while polio drops that are administered into the body contains what? NEET 2016 question. And with this, we complete the topic. I hope that you read or understand the whole concept from your NCERT and then you try to solve the questions. And I hope that the whole thing that I explained was very clear to you. Even I am tired. I know that you might also be tired. So you should be taking a small cutie break. Drink some water, stay hydrated and take care of yourself. In the next class, we are going to talk about AIDS and then cancer and then drug and abuse. Then we are going to complete the whole chapter. That's all. Yeah, so bye bye, take care, and do not forget to subscribe to the channel for the PDF. The link is there in the description box. Bye bye.